Right now, at a depth of 1,000 meters beneath the surface of the Pacific Ocean, a fish is hunting in complete darkness. It has no eyes that function in the way yours do. It does not need them. The darkness around it is not an obstacle. It is a tool, and this fish has spent millions of years learning exactly how to use it. The deep ocean is not empty. It is not silent. It is not waiting. It is full of creatures that evolution has built for conditions so extreme that they seem, by every standard we apply to life at the surface, to be impossible. And yet they exist, they hunt, they reproduce, they compete, they thrive in a world that has no sunlight, no warmth, no mercy. What biology does under those conditions is the subject of this documentary. And what biology does, as it turns out, is something extraordinary. If you enjoy drifting into the deep ocean and discovering what lies beneath, please consider liking and subscribing. It truly helps more than you know. Are you ready? Let's begin. The problem with living in the deep ocean, if you are an animal, is light, specifically the absence of it. Below 200 meters, sunlight does not penetrate. The photons that power every plant, every alga, every photosynthetic organism on the surface of the earth, the particles of energy that ultimately sustain almost all of the life we are familiar with, do not reach the deep ocean. They are absorbed by the water above, scattered and weakened with every meter of depth, until at 200 meters there is nothing left. Below that boundary, the ocean is dark, not dim, not dusky. Dark in the absolute sense, a darkness in which no amount of waiting will allow your eyes to adjust, because there is nothing for them to adjust to. For an animal that evolved in sunlight, this is a problem of the first order. Vision, the primary sense of most vertebrates, the sense that humans rely on more heavily than any other, the sense that shapes our perception of the world more completely than touch or smell or hearing, becomes useless. The eyes that took millions of years to evolve, the neural architecture that processes visual information and turns it into a model of the surrounding world, the behaviors that depend on seeing prey and predators and mates, all of it becomes irrelevant. The deep ocean strips away the most fundamental tool that surface animals use to navigate their environment. And then it offers something in return. Bioluminescence is the production of light by living organisms through chemical reactions. It is not a deep-sea phenomenon exclusively fireflies produce it. Certain species of jellyfish produce it. Some fungi and bacteria produce it in shallow water. But in the deep ocean, it is ubiquitous. Studies using sensitive light-detecting instruments lower through the water column have found bioluminescent flashes at virtually every depth below the photic zone. Estimates suggest that somewhere between 70 and 90% of deep-sea organisms are capable of producing light. In the absolute darkness of the abyss, life has reinvented the sun not as a source of energy, but as a means of communication, predation, defense, and reproduction. The chemistry of bioluminescence is elegant in its simplicity. It involves a molecule called luciferin, which reacts with oxygen in the presence of an enzyme called luciferase to produce a photon of light. The reaction is almost perfectly efficient. Nearly all of the chemical energy invested in it is converted to light, with almost no heat produced. This is in stark contrast to the incandescent bulbs that humans used for a century to light their homes, which convert only a small fraction of their electrical energy to visible light and waste the rest as heat. Evolution, working on the problem of producing light in a cold, energy-scarce environment, arrived at a solution far more efficient than anything human engineering produced until the development of LEDs. The colors of bioluminescence are not random. Most deep-sea bioluminescence is blue or blue-green wavelengths in the range of 470 to 500 nanometers. This is not coincidence. Blue light travels further in water than any other wavelength. Red light is absorbed within the first few meters. Green and yellow light travel somewhat further. Blue light can travel tens of meters through clear ocean water before being absorbed. The organisms that produce blue light are not doing so arbitrarily. They are producing the wavelength that their environment transmits most efficiently. Evolution has tuned the chemistry of deep-sea bioluminescence to the optical properties of the medium through which the light must travel. The anglerfish is the most famous example of bioluminescent predation, and deservedly so. The females of many anglerfish species carry a specialized structure called the esca, a fleshy lure that protrudes from the head on a modified dorsal spine. The esca glows with a steady blue-green light produced by symbiotic bacteria living within it. The anglerfish does not produce the light itself. It farms it, maintaining a colony of luminescent bacteria in a specialized organ, providing them with nutrients and oxygen in exchange for the light they produce. The lure dangles in front of the anglerfish's mouth, waving gently in the current, mimicking the movement of a small organism. What approaches the light any organism curious about or attracted to the glow in the surrounding darkness enters a killing zone. The anglerfish's teeth are long, curved inward, and transparent. They are invisible in the dark water. The jaw can open wide enough to engulf prey larger than the fish's own body, and it can do so faster than most prey can react. In a world where food is scarce and encounters between predator and prey are rare, the anglerfish has evolved a strategy of maximum passivity combined with maximum lethality. It does not hunt, it waits, it glows, and it kills. The dragonfish takes a different approach. These are elongated, slender fish of the deep mesopelagic zone, the region between 200 and 1,000 meters, sometimes called the twilight zone, because faint traces of sunlight penetrate to its upper reaches. Dragonfish have evolved bioluminescence, not just for predation, but for something more sophisticated, private communication. Many species of dragonfish can produce red light, a wavelength that most deep-sea organisms cannot see, because their visual systems are tuned to the blue-green wavelengths that travel furthest in water. The dragonfish, however, can see red light. They have evolved visual pigments sensitive to wavelengths that are invisible to their prey and to most of their predators. The result is a private channel of communication, a light frequency that functions as an invisible wavelength in the visual spectrum of every other organism in the environment, but that is fully visible to the dragonfish themselves. They can signal to one another, navigate by each other's light, and perhaps communicate information about prey or mates, all while remaining effectively invisible to every other organism in the vicinity. The deep ocean did not eliminate light as a medium of communication. It created conditions in which light became a more sophisticated medium than it is at the surface, a medium with hidden frequencies that only certain organisms can access. The siphonophore represents perhaps the most extraordinary deployment of bioluminescence in the deep ocean. Siphonophores are colonial organisms. They are not individual animals, but rather colonies of specialized individual units called zooids, each performing a specific function, collectively constituting what functions as a single organism. The largest siphonophores can reach lengths of 40 meters or more, making them among the longest organisms on Earth. In the deep ocean, certain siphonophores deploy bioluminescence in patterns along their length that may mimic the appearance of small schooling fish, creating a false image of prey that attracts larger predators into the siphonophores. Tentacles. The colony is simultaneously the bait and the trap, a lure measured in tens of meters, hanging in the darkness and waiting. The deep ocean has produced bioluminescent systems of extraordinary diversity and sophistication, precisely because darkness creates the conditions in which light has maximum value. At the surface, light is everywhere. It provides information, but because it is ubiquitous, it does not provide exclusive information. Any organism can see any other organism that produces or reflects light if it has the visual system to detect the relevant wavelengths. In the deep ocean, light is rare and therefore precious. An organism that can produce it has a tool that no other organism in the environment possesses, a tool that can be used to attract, to deceive, to communicate, to warn, or to hide. Hiding with light is a counterintuitive concept, but the deep ocean has produced organisms that do exactly this. Counter-illumination is a strategy employed by many mesopelagic fish and invertebrates, organisms that live in the twilight zone where faint sunlight from above creates silhouettes visible from below. A predator lurking in deeper water can detect prey by looking upward toward the faint light and spotting the dark silhouette of an organism moving through the illuminated water above. The prey has solved this problem by producing light on its underside, matching the intensity and color of the light coming from above, eliminating the silhouette and effectively becoming invisible when viewed from below. The precision required for counter-illumination is remarkable. The organism must measure the intensity and color of the light above it and produce light on its underside that exactly matches these parameters. Too little light, and the silhouette remains visible. Too much light, and the organism becomes a bright spot against a darker background, equally detectable. The system must adjust continuously as the organism moves through water of varying optical properties, as the light from above changes with the angle of the sun, as the depth changes. This is not a simple biological trick. It is a sophisticated sensory and motor system, evolved specifically to solve the problem of visibility in an environment where being seen can mean being eaten. The photophores, light-producing organs that perform counter-illumination in species like the hatchetfish, are distributed precisely across the ventral surface of the fish, oriented downward, and connected to a control system that samples the ambient light and adjusts the output accordingly. The hatchetfish has, in effect, an active camouflage system that operates in the light spectrum, using bioluminescence as a tool not for attraction or communication, but for concealment. What evolution has done with bioluminescence in the deep ocean is use a single basic chemical mechanism, the reaction of luciferin with oxygen, to produce an almost unlimited variety of ecological solutions. The same chemistry powers the anglerfish's lure and the dragonfish's private frequency and the siphonophore's false school and the hatchetfish's counter-illumination. Each application solves a different problem. Each represents a different evolutionary path taken from the same starting point. The deep ocean is dark, and darkness is a problem, and life has found more solutions to that problem than we have yet fully cataloged. There is one more aspect of deep-sea bioluminescence that deserves particular attention, because it reveals something about the nature of evolution in extreme environments that is easy to overlook. Bioluminescence has evolved independently in deep-sea organisms many times, not once, not twice, but dozens of times across dozens of unrelated lineages. Fish, squid, shrimp, jellyfish, siphonophores, bacteria, all have independently arrived at the same solution to the problem of darkness. The specific chemistry varies somewhat between lineages. The specific organs that produce the light differ in their structure. The specific uses to which the light is put vary across species. But the basic solution, make your own light, has been discovered again and again, in lineage after lineage, over hundreds of millions of years of evolution in the deep ocean. This convergence tells us something important. It tells us that bioluminescence is not an unlikely or difficult evolutionary innovation. It tells us that, given the right selective pressure, the right problem demanding a solution, evolution finds the solution reliably. The deep ocean provides a clear and consistent selective pressure. Darkness is a problem, and light is a solution. And any organism that can produce light has a substantial advantage over organisms that cannot. The result, repeated across dozens of lineages, is the same: light production. The deep ocean has run the experiment of evolution under conditions of absolute darkness many times over, in many separate lineages, and it has gotten the same answer every time. The implication is that bioluminescence, wherever conditions demand it, will emerge. It is not a cosmic accident, not a unique evolutionary event, not something that happened once on Earth and might or might not have happened elsewhere. It is a predictable outcome of evolution under specific selective pressures. If there are organisms in the dark subsurface oceans of Europa or Enceladus, and we do not know whether there are, they may have solved the problem of darkness the same way that Earth's deep-sea organisms have solved it, by making their own light. The deep ocean has shown us that when evolution confronts darkness, it reaches for chemistry. It finds luciferin and luciferase, or something chemically equivalent, and it makes light The darkness of the deep ocean is not a barrier to life. It is a workshop, a place where over hundreds of millions of years, evolution has refined its tools for operating in the absence of sunlight and produced organisms of extraordinary sophistication and diversity. What bioluminescence has become in the deep ocean, the variety of its forms, the precision of its regulation, the ingenuity of its applications is a testament to what evolution can accomplish when it has enough time and a clear enough problem to solve. The darkness, in the end, is not what defines the deep ocean. It is what the deep ocean has done with the darkness that matters, and it has done more with it than we have yet fully understood. The counter-illumination strategy of the hatchetfish points to something broader about how the deep ocean has shaped animal sensory systems. Eyes in the deep ocean are not the straightforward organs they are at the surface. The problem of seeing in near total darkness has driven some of the most remarkable sensory innovations in the animal kingdom. Innovations that reveal, in their diversity and their precision, just how powerful a selective pressure the absence of light can be. The barrel-eye fish is perhaps the most striking example. This small deep-sea fish has transparent eyes, not transparent in the sense of being simply clear, but transparent in the most literal sense. You can see through the fish's head to the organs within. The eyes themselves are large, tubular structures pointing upward, maximizing the collection of the faint, downwelling light from above. They are not fixed in the skull. They can rotate, allowing the fish to look forward when pursuing prey or upward when scanning for the silhouettes of organisms above it against the faint light of the surface. The barrel-shaped structure of the eye giving the fish its name concentrates available light onto a large retina, maximizing sensitivity at the cost of a narrow field of view. For decades, researchers who caught barrel-eye fish in nets believed that the greenish, dome-shaped structures on the front of the fish's head were its eyes. They are not. They are olfactory organs, structures for detecting chemicals in the water. The actual eyes, inside the transparent head, were not recognized as such until researchers used a remotely operated vehicle to observe living barrel-eye fish in their natural environment and watch the eyes rotate within the transparent dome of the skull. The fish had been studied for a century, and its most distinctive feature had been misidentified for that entire time. The deep ocean hides its secrets, even from scientists who think they are looking directly at them. The giant squid presents a different solution to the problem of seeing in the deep ocean. These organisms, among the largest invertebrates on Earth, reaching lengths of 13 meters or more, have the largest eyes of any animal known to science. A single eye of a large giant squid can reach a diameter of 27 centimeters, roughly the size of a soccer ball. No other animal approaches this. The function of such enormous eyes in the deep ocean has been debated, but the leading hypothesis is that they are tuned not to the faint ambient light of the deep, but to the bioluminescent flashes produced by other organisms. A predator moving through the water produces bioluminescent flashes as it disturbs organisms in its path. The giant squid's enormous eyes may be specialized to detect these flashes, to see not the predator itself, but the light that the predator's movement generates in the surrounding water. The predator in question, most likely, is the sperm whale. These are the primary predators of giant squid in the deep ocean, and they are large enough and active enough that their movements through the water would produce detectable bioluminescent flashes across considerable distances. A giant squid with eyes capable of detecting these flashes at distances of tens of meters would have time to react, to flee, to take evasive action, to avoid the whale before it arrived. The arms of the giant squid bear the scars of sperm whale suction cups, and the stomachs of sperm whales contain the beaks of giant squid. These two organisms have been in an evolutionary arms race for millions of years, and the giant squid's eyes, 27 centimeters in diameter, larger than any other animal's, may be the result of that race. This is what the deep ocean produces. Evolutionary arms races fought in the dark with weapons made of light and silence and patience. The anglerfish's lure versus the prey animal's caution. The dragonfish's private light frequency versus the visual systems of its competitors. The giant squid's enormous eyes versus the sperm whale's echolocation. The counter-illuminating hatchetfish versus the upward-looking predator below. In each case, the darkness of the deep ocean has not simplified the biological interactions that take place there. It has made them more complex, more refined, more precisely calibrated to the specific conditions of an environment where light is rare and food is scarce and every adaptation matters. The deep ocean is not a simplified world. It is not a world of primitive, undifferentiated organisms surviving at the margins of existence. It is a world of extraordinary biological sophistication, shaped by pressures that surface life never experiences, solved by evolutionary innovations that surface biology never needed to develop. What the deep ocean has done with light, with the presence of bioluminescence and the absence of sunlight, is only one example of what it has done with the extreme conditions of its environment. The darkness is where this story begins. It is not where it ends. Pressure is the defining physical reality of the deep ocean. Not darkness. Darkness is an absence, a lack of something. Pressure is a presence. It is real. It is measurable. It is everywhere. And at the depths where the organisms in this documentary live, it is crushing in a way that the word barely captures. At 1,000 meters, the pressure is 100 atmospheres, 100 times the weight of the air pressing down on you right now. At 4,000 meters, 400 atmospheres. At the bottom of the Mariana Trench, over 1,000 atmospheres. This is not a hostile environment in the way that a desert is hostile or a polar ice cap. It is hostile in a way that makes those environments seem accommodating. The deep ocean does not merely challenge life. By every physical standard we can apply, it should prevent it. And yet, the organisms that live there are not struggling. They are not clinging to existence at the margins of what biology permits. They are doing what organisms do everywhere, competing, evolving, finding food, reproducing, dying, and being replaced by organisms slightly better adapted than the ones before them. The pressure that should make them impossible has instead made them extraordinary. Understanding how requires looking at what pressure actually does to living tissue, and then understanding what evolution has done over hundreds of millions of years to undo it. The fundamental problem with pressure is what it does to proteins. Proteins are the molecular machines of life, the enzymes that catalyze every chemical reaction in a cell, the structural molecules that give cells their shape, the receptors that allow cells to sense and respond to their environment. Proteins function because they fold into specific three-dimensional shapes, and those shapes are maintained by relatively weak chemical interactions, hydrogen bonds, hydrophobic effects, van der Waals forces. At the pressures of the deep ocean, these weak interactions are disrupted. Proteins unfold, enzymes lose their catalytic activity. Cell membranes, which are assemblies of lipid molecules, become compressed and rigid, losing the fluidity that allows membrane proteins to function. The biochemistry of life, tuned over billions of years of evolution at surface pressures, breaks down. The solution that deep-sea organisms have evolved to this problem is molecular. Their proteins have different amino acid sequences from the proteins of their surface relatives, not dramatically different, but different in ways that matter. The substitutions are often at positions that affect the flexibility of the protein structure, making the proteins more rigid in ways that resist the deforming effects of pressure. Deep-sea enzymes typically function at their optimal rate at the pressures where the organisms that carry them live. The same enzymes, when tested at surface pressures, are often less active because their structure is too rigid for the low-pressure environment. They are not adapted for the surface. They are adapted for the deep. Cell membranes in deep-sea organisms show a parallel adaptation. The fluidity of a membrane depends on the types of lipids it contains, specifically on the degree of unsaturation of the fatty acid chains in the membrane phospholipids. More unsaturated fatty acids make a membrane more fluid. Deep-sea organisms tend to have higher proportions of polyunsaturated fatty acids in their membranes than their shallow water relatives, an adaptation that maintains membrane fluidity at the temperatures and pressures of the deep ocean. These are the same omega-3 fatty acids that nutritionists recommend as components of a healthy human diet. The reason they appear in deep-sea fish at high concentrations is not coincidental. It is an evolutionary adaptation to maintain membrane function in extreme conditions. The blobfish is perhaps the most notorious example of pressure adaptation in popular culture, and also perhaps the most misunderstood. The blobfish, as most people have seen it, is a grotesque, sagging mass of pale flesh with a drooping face that has become one of the most widely shared images in internet culture. What almost no one who sees that image understands is that the blobfish does not look like that in its natural environment. The image of the sagging, melting face is the result of decompression, of bringing the fish from the deep ocean to the surface, where the pressure that maintained the structure of its tissues drops from dozens of atmospheres to one. The decompression causes the gases dissolved in the fish's body to expand, the fluids to redistribute, the structural integrity of the tissues to collapse. The blobfish you see in photographs is not a living animal in any functional sense. It is an organism that has been subjected to the equivalent of explosive decompression pulled from the only pressure conditions under which its biology makes sense. In its natural environment, at depths of 600 to 1200 meters, the blobfish is a relatively normal-looking fish. Its flesh is slightly gelatinous compared to surface fish, a property that helps it maintain neutral buoyancy at depth without a gas-filled organ that would be crushed at those pressures. It moves slowly across the seafloor, consuming whatever organic matter it can find. It is not grotesque. It is simply adapted to conditions that destroy it when removed. The grotesque image of the blobfish is an image of what pressure adaptation looks like when you remove the pressure, a reminder that deep-sea organisms are not bizarre by nature. They are bizarre only when taken out of the context that makes their biology coherent. The structural adaptations of deep-sea organisms extend to their skeletons. Many deep-sea fish have reduced skeletons' lighter bones, less calcified, more cartilaginous than their shallow water relatives. This is partly an energy conservation strategy. Building and maintaining bone is metabolically expensive, and in an energy-scarce environment, any reduction in metabolic cost has value. But it is also a pressure adaptation. Lighter, more flexible skeletal structures are better suited to the compressive forces of the deep ocean than the rigid, heavily calcified skeletons of surface fish. The deep ocean selects against rigidity and for flexibility, a principle that applies not just to skeletons, but to every structural component of the organisms that live there. The giant isopods illustrate a different aspect of pressure adaptation: gigantism. These crustaceans, relatives of the pill bugs that curl into balls in garden soil, can reach lengths of 45 centimeters or more in the deep ocean, far exceeding the size of any shallow water relatives. Giant isopods, giant amphipods, giant sea spiders. The pattern of deep-sea gigantism appears repeatedly across unrelated lineages. The explanations are debated. Cold temperatures slow metabolic rates, which may allow animals to grow larger before reproduction, the cold temperature gigantism hypothesis. Low oxygen partial pressure may paradoxically allow larger body sizes in some lineages. The absence of competitors at certain size ranges may allow organisms to occupy niches unavailable in the more crowded shallow ocean. Whatever the cause, deep-sea gigantism is real and consistent. The organisms of the deep ocean are not uniformly small and compressed by their hostile environment. Many of them are among the largest representatives of their groups anywhere on Earth. The deep ocean does not merely tolerate size in some cases, it produces it, selecting for organisms large enough to survive the long intervals between food pulses that characterize the abyssal environment. The transparent organisms of the deep ocean represent yet another suite of pressure adaptations working in concert with other environmental demands. Many deep-sea jellyfish, cenophores, and small crustaceans are nearly or completely transparent. Their tissues allow light to pass through them without absorption or scattering. Transparency is, in the deep ocean, a form of camouflage. An organism that is transparent has no silhouette, no color, no visual signature. It is effectively invisible in an environment where vision is already limited. The pressure adaptations required for transparency are significant. Transparent tissue must be maintained under conditions that would normally cause proteins to unfold and cell membranes to lose their structure. But the camouflage benefit is sufficient to have driven the evolution of transparency across dozens of unrelated deep-sea lineages. The fang-toothed fish illustrates what the scarcity of food in the deep ocean does to feeding apparatus. This small deep-sea fish rarely longer than 15 centimeters, has the largest teeth of any fish in the ocean relative to its body size. The fangs are so long that the fish cannot close its mouth completely. The lower teeth slot into specialized pockets in the upper jaw to prevent them from puncturing the brain when the mouth shuts. The fanged tooth does not hunt actively in the way that surface predators do. It cannot afford the energy expenditure of sustained pursuit. Instead, it drifts through the water column, its enormous teeth forming a cage from which prey cannot escape once contact is made. In a world where the next meal may be hours or days away, a jaw that closes around prey and does not release it is worth the cost of never being able to shut one's mouth entirely. The gulper eel takes feeding adaptation to an extreme that seems to border on biological satire. This organism is largely mouth and stomach. Its body is a long, slender tube terminating in a small tail. Its head is dominated by an enormous, loosely articulated jaw that can open to swallow prey many times the eel's own diameter. The jaw is essentially a net. When the gulper eel encounters prey, it opens its mouth and swims forward, engulfing whatever is in front of it. The stomach expands to accommodate the meal. The eel then digests slowly, processing the food over hours or days, while its body accommodates a bolus of material that, in terms of mass, may exceed its own body weight. The viperfish has evolved a different solution to the scarcity problem. Its teeth, long, curved, needle-like structures that protrude from its closed mouth, are so large relative to its head that the fish cannot close its jaws completely. The teeth interlock when the mouth is shut, forming a cage that prevents prey from escaping once caught. The viperfish is not selective about what it catches. It cannot afford to be. In an environment where the next meal may be hours or days away, and where that meal may be moving fast enough to escape if not immediately secured, the viperfish has evolved teeth that function as a trap rather than simply as weapons. These feeding adaptations, the fanged tooth's interlocking teeth, the gulper eel's enormous jaw, the viperfish's cage of fangs, share a common logic. They are responses to the same fundamental pressure. Food in the deep ocean is scarce, unpredictable, and potentially large relative to the predator. An organism that can exploit food items across a wide range of sizes, that can consume more than it immediately needs, that can secure prey quickly and hold it reliably. Such an organism has a survival advantage over one constrained by the limitations of a surface fish's body plan. The deep ocean does not reward elegance. It rewards capacity. The deep ocean body plans that result from this selection pressure are, by surface standards, often grotesque. The gulper eel's enormous mouth, the viperfish's protruding teeth, the blobfish's gelatinous flesh, the fanged tooth's impossibly oversized fangs. These features look like deformities, like evolution gone wrong, producing organisms that are more caricature than creature. But they are not deformities. They are solutions. Each feature solves a specific problem posed by the deep ocean environment, solves it efficiently, and has been maintained by natural selection because organisms with it survive and reproduce better than organisms without it. What the deep ocean looks like, from the perspective of surface biology, is a chamber of evolutionary horrors. A place where the normal rules of body plan and feeding strategy have been abandoned in favor of something alien and strange. What it actually is, from the perspective of evolution, is a particularly clear demonstration of what natural selection does when given extreme selective pressures and sufficient time. It finds solutions. It finds them reliably. And the solutions it finds are exactly as strange as the problems they solve. No stranger, no less strange, but precisely as strange as the environment demands. There is one more dimension of pressure adaptation that connects the biology of individual organisms to the broader ecology of the deep ocean. The pressure adaptations of deep-sea organisms are not merely individual survival strategies. They are the foundation of an ecological community, a set of biological capabilities that, taken together, make the deep ocean a functional ecosystem rather than a collection of isolated survivors. Consider what it means for the deep ocean ecosystem that many of its organisms have no gas-filled buoyancy organs, structures that would be crushed to nothing at depth. Deep-sea fish that lack these organs must achieve neutral buoyancy through other means: reduced calcification of the skeleton, high water content in the tissues, high concentrations of lipids, which are less dense than water. These alternatives are less precise, but they work at pressures where gas-filled organs cannot. And crucially, they allow vertical movement, without a rigid buoyancy organ constraining their depth range. These fish can migrate upward into shallower water at night to feed in the relatively richer waters of the mesopelagic zone, then descend during the day to avoid predators in the better-lit upper layers. This dio-vertical migration is one of the most significant biological processes in the ocean. The organisms that perform it carry nutrients and organic matter from the surface ocean to the deep, transferring energy downward in ways that support the entire deep ocean food web. A pressure adaptation, the elimination of a rigid buoyancy organ, makes this migration possible, and the migration sustains a community of organisms that would otherwise starve. The deep ocean is not a simple environment with simple organisms. It is an ecosystem of extraordinary complexity, in which physical conditions have shaped biological adaptations that in turn shape ecological processes that in turn sustain the entire community. Pressure is not merely a constraint. It is a sculptor, the force that has shaped deep-sea biology into the forms it takes, and in doing so, created the ecological framework within which those forms can survive. The organisms of the deep ocean are not accidents. They are the products of hundreds of millions of years of evolution in one of the most extreme environments on Earth, environments stable enough and persistent enough for evolution to find solutions that work. The solutions it has found are functional in a deep sense. They solve problems that surface biology never faces, in conditions that surface biology never encounters, with a precision that becomes apparent only when you understand the problem being solved. Darkness, pressure, cold, scarcity, these are the parameters of the problem. And what evolution has built in response is not a world of monsters. It is a world of answers. And the next category of answers is perhaps the most alien of all. The black smoker chimneys of the hydrothermal vent systems represent pressure adaptation taken to its absolute thermal extreme. Bacteria and archaea, the ancient single-celled life forms that predate all complex multicellular life, have colonized the walls of these chimneys. Living in the zone where water at 300 or 400 degrees Celsius meets the cold ambient ocean, their proteins function at temperatures that denature the proteins of every other known organism. Their membranes maintain their integrity at temperatures that melt the membranes of ordinary cells. They do not merely tolerate these temperatures, they require them. Cooled to ordinary biological temperatures, they die. The discovery that life can function at temperatures above the boiling point of water at surface pressure, that the biochemistry of proteins and membranes can be adapted to conditions that should make them impossible extended. The known range of life, as dramatically as the discovery of chemosynthesis, extended the known range of energy sources. Before these organisms were found, the upper temperature limit for life was thought to be around 70 or 80 degrees Celsius. The black smoker archaea pushed that limit past 120 degrees. And with each upward revision of the thermal limit, the range of environments in the universe that could, in principle, support life expanded correspondingly. The pressure adaptations of the deep ocean have, in this way, done something that extends far beyond the organisms themselves. They have demonstrated that life is more physically flexible than we assume, that the constraints on where biology can function are less absolute than the conditions of the surface world suggested. Every organism that thrives at 1,000 atmospheres of pressure, every archaean that lives above the boiling point of water, every transparent jellyfish that maintains its cellular integrity at temperatures near freezing each, is a data point in an argument about what life requires. And the argument, taken together, is that life requires less than we thought and can adapt to more than we imagined. This matters for how we think about the rest of the universe. The conditions of the deep ocean, high pressure, cold temperature, complete darkness, chemical rather than solar energy, are not exotic in a cosmic sense. They are common. The frozen moons of the outer solar system, harboring liquid water beneath their ice, may have conditions not unlike the deep ocean floor. Dark, cold, pressurized, potentially rich in the chemical energy that hydrothermal activity provides. The organisms of the deep ocean have shown that life can thrive in exactly such conditions. They have not proven that life exists elsewhere. But they have made the question worth asking, and they have pointed to the kinds of environments where the answer might be yes. The deep ocean's pressure adaptations are, in this light, not merely biological curiosities. They are evidence. Evidence about the nature of life, about what it requires and what it can endure, about the range of conditions under which the chemistry of self-replication and evolution can persist. The organisms that carry these adaptations do not know that they are making an argument. They simply live, as they have always lived, in the conditions that shape them. But the argument they make, for anyone willing to read it, is one of the most important arguments in biology. The deep ocean has been making this argument for hundreds of millions of years. We have been listening for less than a century, and we are only beginning to understand what it is saying. Chemoreception, the detection of chemicals dissolved in the surrounding medium, is the oldest sense in the animal kingdom. Single-celled organisms that lived in the primordial ocean more than 3 billion years ago could detect chemical gradients and move toward or away from them. Every complex animal that has evolved since carries some version of this ancient capability. In the deep ocean, where light fails and sound becomes the primary alternative, chemoreception has been refined to a degree that surface organisms relying primarily on vision have never needed to develop. The hagfish is perhaps the most striking example of deep-sea chemoreception taken to an extreme. These are among the most ancient vertebrates on Earth. Their lineage extends back at least 300 million years, largely unchanged. A persistence that itself tells you something about how well their biology suits their environment. Hagfish have no true eyes. The structures that remain are vestigial, capable of detecting the difference between light and dark, but not of forming images. They have no jaw in the conventional sense. They are, by most visual assessments, not impressive animals. What they have is a chemosensory system of extraordinary sensitivity. The hagfish can detect the chemical signature of a decaying carcass from enormous distances and the specific mixture of amino acids, sulfur compounds, and organic molecules released by decomposing flesh diffuses through the water column over hours and days, creating a gradient that the hagfish follows to its source. In the featureless darkness of the deep ocean, where a whale carcass represents months of food, the ability to detect that carcass from a kilometer away is not a minor advantage. It is the difference between survival and starvation. The hagfish does not simply smell the carcass, it enters it. Using its specialized oral apparatus, a circular arrangement of tooth-like structures that grip and tear it burrows into the flesh of the dead animal, surrounding itself with food, consuming it from within. When threatened, it releases enormous quantities of slime, a mucus gel of extraordinary volume and viscosity that clogs the gills of any fish that tries to bite it. The slime is itself a chemically sophisticated substance. It expands in water to volumes many times larger than the hagfish that produced it, and its structure is maintained by protein fibers of remarkable tensile strength. The hagfish did not evolve to be impressive in the way that surface predators are impressive. It evolved to find food in the dark, enter it, and defend itself when challenged. It has been doing these things, essentially unchanged, for 300 million years. The lateral line system represents a different category of deep-sea sensory adaptation, one that detects not chemistry, but the physical movement of water. Fish have long been known to possess a lateral line, a series of sensory organs running along the length of the body that detect pressure changes and water movements in the surrounding medium. At the surface, the lateral line is a useful supplement to vision and hearing, allowing fish to detect the movements of nearby prey or predators in conditions of reduced visibility. In the deep ocean, where visibility is zero, the lateral line becomes a primary sense. Deep-sea fish have lateral line systems of considerably greater sensitivity and resolution than their surface relatives. The individual sensory units, called neuromasts, are more numerous, more densely distributed across the body surface, and more sensitive to small pressure changes. Some deep-sea fish have evolved canal systems that concentrate and amplify the water movements detected by the neuromasts, increasing their effective range. The result is an organism that can detect a pressure wave produced by a small fish swimming at a distance of several meters, a signal so faint that it would be lost against the background noise at the surface, but that is detectable. In the quieter, deep ocean, the relationship between sound and the deep ocean is more complex than most people realize. Water is an excellent medium for sound transmission. Sound travels roughly 5 times faster in water than in air and loses energy much more slowly with distance. In the surface ocean, this means that whales can communicate across thousands of kilometers, their calls propagating through the water with remarkable efficiency. In the deep ocean, there is a specific depth range, roughly between 600 and 900 meters in most ocean basins, varying with temperature and salinity, called the sulfur channel, where the speed of sound reaches a local minimum. Sound produced at this depth tends to be refracted back toward it by the layers of water above and below, creating a natural acoustic waveguide that can carry sound signals across entire ocean basins. Deep-sea organisms have not ignored this physics. Some deep-sea fish produce sounds, low-frequency clicks, grunts, and pulses that may propagate through the sulfur channel and serve as long-range communication signals. The mechanisms of sound production in these fish are often novel. Rather than using the gas-filled organs that many surface fish use to amplify sounds, deep-sea fish have evolved alternative sound-producing structures. Some use modified tendons that vibrate when muscles contract. Others use specialized bones that rub against each other. The sounds produced are not songs in the way that whale songs are songs. They do not appear to have the complex structure that suggests elaborate communication. But they are intentional signals, produced at specific times and in specific contexts, and they carry information through the water to organisms that can detect them. The detection of these low-frequency sounds requires a different kind of hearing organ than the one used to detect the higher frequencies that dominate surface environments. Many deep-sea fish have evolved otoliths, dense calcium carbonate structures in the inner ear that are disproportionately large relative to their body size. Larger otoliths are more sensitive to lower frequencies and to smaller displacements, making them better suited to detecting the faint, low-frequency signals that propagate through the deep ocean. This is not a random variation in anatomy. It is an adaptation to the acoustic environment of the deep ocean, a sensory system tuned to the frequencies that carry furthest in the medium where the organism lives. The sense of electroreception, the detection of electric fields, is rare among vertebrates, but it is present in some deep-sea species and may be particularly valuable in the deep ocean environment. Electroreception was a common feature of early vertebrates, and it has been retained in some lineages, lost in others. In sharks and rays, including the deep-sea species that inhabit the ocean floor, the ampullae of Lorenzini are electroreceptive organs that can detect the weak bioelectric fields produced by the muscle contractions of other organisms. At very short ranges, this allows a predator to detect prey that is visually invisible, acoustically silent, and chemically undetectable prey that has successfully hidden from every other sense the predator possesses. In the deep ocean, where prey animals have had millions of years to evolve, concealment, transparency, bioluminescent camouflage, silence, minimal chemical emission, the ability to detect the bioelectric field of a muscle contraction is a sense that cannot be evaded by any passive means. Every organism with muscles produces bioelectric fields. These fields cannot be suppressed without suppressing the muscle contractions themselves, which would require the organism to stop moving entirely. Electroreception is the sense that sees through concealment, that detects life itself rather than any secondary signal that life produces. The deep ocean has also shaped chemical communication between organisms in ways that go beyond the detection of food. Many deep-sea organisms release specific chemical signals, pheromones, that carry information about reproductive state, species identity, and location. In an environment where encounters between organisms of the same species are rare and unpredictable, and where those encounters cannot be initiated through visual signals, chemical communication becomes essential for reproduction. A sexually receptive female that cannot signal her condition visually, acoustically, or through proximity, because the darkness and the distances of the deep ocean prevent all three, must rely on chemistry. She releases pheromones into the water, and those pheromones diffuse outward in a gradient that a male with a sufficiently sensitive chemosensory system can follow to its source. One deep-sea species has taken chemical mate finding to its most extreme biological conclusion. The male of this species is a tiny organism, far smaller than the female, and its primary function is reproduction. It hatches from the egg already equipped with an unusually large olfactory organ, a chemosensory system that, relative to its body size, is among the most developed in the animal kingdom. This is the male of one of the deep ocean's most studied families, a fish whose females you have already met, known for their bioluminescent lures, here encountered from an entirely different angle. It spends its early life following female pheromone gradients through the darkness of the deep ocean. When it finds a female, it bites into her flesh and fuses with her body. The male's own circulatory system connects to the female's. His organs, eyes, digestive system, most of his brain atrophy and disappear. He becomes, essentially, a sperm-producing appendage permanently attached to the female, nourished by her blood, existing solely to fertilize her eggs when she produces them. This is reproduction in the deep ocean, a sensory odyssey through a chemical gradient, ending in a permanent fusion that eliminates the need for any further search. He does not need to find another female. He does not need to maintain the sensory systems that found this one. He has arrived. The deep ocean has driven evolution to produce a reproductive system that, by surface standards, seems grotesque, but that is, in the context of the deep ocean, perfectly rational. Encounters between members of the same species are too rare to risk losing. The solution is to make every encounter permanent. The chemical world of the deep ocean extends beyond pheromones and the detection of food and mates. Some deep-sea organisms produce chemicals that are toxic to other organisms, compounds that serve as defenses against predation or as weapons in competition for resources. The chemistry of deep-sea toxins is poorly understood, partly because collecting living specimens from the deep is difficult, and partly because the organisms that produce these compounds are not abundant enough to study in large numbers. But what has been studied suggests that deep-sea chemical ecology is as complex and as sophisticated as the chemical ecology of any other environment. A world of molecular signals, warnings, invitations, and weapons operating in a medium that carries them efficiently and conceals the organisms that produce them. The sensory world of the deep ocean is not impoverished. It is different. Surface organisms live in a world dominated by light, and the information that light carries: color, shape, movement, distance. Deep-sea organisms live in a world dominated by chemistry, and the information that chemistry carries: identity, direction, distance, reproductive state, metabolic activity. These are not lesser sources of information. In many respects, they are richer chemistry, carries more molecular specificity than light, more unique information per signal, more precise identification of source and sender. The deep ocean has not simplified the sensory world of its inhabitants. It has shifted it from the visual and auditory to the chemical and mechanical, from the fast and directional to the slow and diffuse, from the obvious to the hidden. What moves through the deep ocean, through the water, through the sediment, through the bodies of the organisms that live there, is not silence and emptiness. It is information. Chemical gradients and pressure waves and bioelectric fields carrying messages that the organisms of the deep ocean have been reading for hundreds of millions of years. The darkness hides nothing from them. The darkness is simply the medium in which they have learned to read what the light would otherwise obscure. There is a dimension of deep-sea sensory biology that connects all of these systems: chemoreception, lateral line, electroreception, acoustics, in a way that reveals something fundamental about how evolution works in extreme environments. Each of these senses is not a new invention. Each has its roots in the sensory systems of ancient organisms that lived in the surface ocean long before the deep ocean was colonized. What evolution has done in the deep ocean is not create new senses. It has refined, amplified, and repurposed existing ones, turning up the sensitivity of chemical detection, extending the range of mechanical sensing, developing novel applications for electrical field detection. This pattern of refinement, rather than invention, is characteristic of how evolution responds to extreme environments. The organisms that first colonized the deep ocean did not arrive with entirely new biology. They arrived with the biology of their surface ancestors, and they survived because variations in that biology happened to function better in the dark, cold, pressurized environment they encountered. Those variations were selected for. Generations of selection amplified them. And over millions of years, what began as a subtle difference in the sensitivity of an olfactory receptor or the size of an otolith became the extraordinary chemosensory system of the hagfish or the low-frequency hearing of the deep-sea fish. This matters for how we think about the deep ocean's future. The organisms that live there are not static. They are evolving now, as they have always been, responding to the selective pressures of their environment. And the selective pressures of that environment are changing. The deep ocean is warming. It is acidifying. The organic matter rain from above. The marine snow that forms the primary food supply for abyssal organisms is changing in quantity and composition as the surface ocean responds to warming temperatures and altered circulation patterns. The chemical environment of the deep ocean is shifting, slowly but measurably. For organisms whose primary interface with their world is chemical organisms, whose survival depends on detecting specific molecular signals in the surrounding water, these changes in chemistry may have consequences that we cannot fully predict. A shift in the chemical composition of the organic matter, arriving from above, changes what the hagfish can smell and follow. A change in seawater pH changes the propagation of chemical signals through the water. A change in temperature changes the rates at which chemical gradients form and dissipate. The sensory world of deep-sea organisms is built on the chemistry of their environment, and that chemistry is not as stable as it was. We do not know how deep-sea organisms will respond. We know that evolution is capable of responding, that natural selection can, given enough time, adapt a population to new chemical conditions, refine a sensory system to function in a different molecular environment, find new sources of information in a changed world. What we do not know is whether the rate of change will outpace the rate of evolutionary response, whether the deep ocean is changing faster than its inhabitants can adapt. These are not merely academic questions. The sensory systems of deep-sea organisms are, in many cases, more refined than anything we have engineered. The chemical sensing systems of organisms, like the hagfish, can detect concentrations of specific molecules that are orders of magnitude below the detection thresholds of our most sensitive laboratory instruments. The electroreceptive systems of deep-sea sharks can detect electric fields that would be invisible to any instrument we have built. The lateral line systems of deep-sea fish can detect water movements that are beyond the resolution of our most sensitive pressure sensors. Understanding how these systems work, understanding the molecular mechanisms by which they achieve their extraordinary sensitivity, has direct implications for human technology. Biosensors, inspired by deep-sea chemoreception, could detect chemical pollutants, medical biomarkers, or explosive compounds at concentrations that current sensors cannot reach. Materials inspired by the slime of the hagfish could produce remarkably strong, flexible, lightweight textiles. The acoustic processing of deep-sea fish could inform the design of sonar systems that operate more efficiently in challenging acoustic environments. The deep ocean is not merely a place of biological curiosity. It is a 40-million-year research and development laboratory in which evolution has been solving engineering problems under extreme constraints, producing solutions that human engineering has rarely approached. Every organism that lives there is a solution to a problem. Every sensory system is an answer to a challenge. And most of those answers we have not yet read. The chemistry moves through the water in the dark. The pressure waves propagate across kilometers of cold ocean. The bioelectric fields pulse with the contractions of invisible muscles. And in that sensory world, invisible to us, inaudible to us, chemically imperceptible to us, an entire community of organisms navigates, hunts, reproduces, and survives. They do not need our instruments to find each other. They have been finding each other in the dark for longer than our species has existed, and they will continue to do so in the dark long after we are gone, unless the world we are changing makes that continuity impossible. That question whether the biology of the deep ocean can survive what the surface world is doing to it is one that the organisms themselves cannot ask. Only we can ask it. And whether we ask it in time to matter is, as yet, undetermined. Time moves differently in the deep ocean. This is not a metaphor. It is a biological fact. The organisms that inhabit the deep ocean live at metabolic rates so slow that their relationship to time, to growth, to aging, to reproduction, to death is fundamentally unlike anything found in the surface world. A fish that lives for 5 years in a coastal estuary might have a deep ocean relative that lives for 500. A crustacean that reproduces annually at the surface might have a deep-sea counterpart that reproduces once a decade. The cold temperature of the deep ocean slows every biochemical reaction, and the cumulative effect of that slowing, multiplied across an entire lifetime, is a creature that experiences the passage of time at a fraction of the rate that surface organisms do. The orange roughy is perhaps the most studied example of deep-sea longevity in a commercial species. This fish, inhabiting depths of between 100 and 1,800 meters in the southern oceans, can live for more than 150 years. It does not reach sexual maturity until it is roughly 30 years old. A female orange roughy that is caught and sold as seafood may be older than a human great-grandmother, and she may never have reproduced because she had not yet reached the age at which reproduction begins. The commercial fishing industry did not know this when it began harvesting orange roughy in the 1970s. The fish were abundant. They schooled in ways that made them easy to catch in large numbers, and they were delicious. The industry expanded rapidly. What the industry did not know, because the biology of the fish was not yet understood, was that an orange roughy population recovers from exploitation far more slowly than a surface fish population. A cod population that has been overfished may recover in a decade. An orange roughy population that has been overfished may take a century to recover if it recovers at all. By the time this was understood, many orange roughy populations had been reduced to fractions of their former size, and some have never recovered. The orange roughy story is a cautionary tale about what happens when surface assumptions are applied to deep-sea biology. The assumption that a fish that reproduces slowly and lives long is rare at the surface. Most commercially harvested surface fish are fast-growing, early maturing, short-lived species. The deep ocean, with its slow metabolism and long lifespans, inverts this, and the inversion matters enormously for conservation. A deep-sea species cannot be harvested at rates that would be sustainable for a surface species. The mathematics of population recovery simply do not allow it. The orange roughy is not an outlier in the deep ocean. It is representative of a pattern that appears across taxa. Deep-sea corals, colonial organisms related to the tropical corals of shallow reefs, but inhabiting the cold, deep waters of the continental slopes and seamounts, can live for thousands of years. A single colony of a deep-sea coral species called Leopathus has been dated at over 4,000 years old, one of the oldest living organisms on Earth, anchored to a seamount in the Pacific, growing at a rate of micrometers per year. It was alive before the pyramids of Egypt were built. It will presumably still be alive if undisturbed when whatever comes after our civilization has risen and fallen. What does it mean to be alive for 4,000 years? For the coral, it means very little in experiential terms. A coral colony has no nervous system, no subjective experience, no memory. But in biological terms, it means something remarkable. It means that this colony has been building its calcium carbonate skeleton, feeding on the particles drifting past it in the current, withstanding the occasional physical disturbance of the deep ocean for 4 millennia. It has weathered changes in ocean temperature and chemistry that have occurred over that period. It has survived whatever episodic disturbances, turbidity currents, changes in circulation patterns, fluctuations in food supply have affected its particular location on the seamount. Its longevity is itself an adaptation. A colony that lives for thousands of years does not need to reproduce frequently to maintain its population, and the energy that would otherwise go into reproduction can go instead into continued growth. The relationship between metabolism and lifespan in the deep ocean is not simply that slow metabolisms produce long lives. It is more precisely that slow metabolisms reduce the rate of cellular damage, the accumulation of oxidative stress, the degradation of DNA repair mechanisms, the progressive dysfunction of cellular processes that eventually leads to the death of the organism in every species that has been studied. The biochemistry of aging is not fully understood, but what is understood suggests that the rate of aging is closely tied to the rate of metabolic activity. Deep-sea organisms, metabolizing at a fraction of the rate of surface organisms, may age correspondingly more slowly and may therefore live correspondingly longer. This has implications that extend beyond deep-sea biology. The study of aging is one of the most active fields in biomedical research, and deep-sea organisms represent a largely unexplored resource for understanding how aging can be slowed or modified. The proteins that maintain cellular integrity at extreme pressures, the lipid compositions that keep membranes functional at near freezing temperatures, the DNA repair mechanisms that allow organisms to persist for thousands of years in a radiation-free but chemically challenging environment. Any of these could contain insights applicable to human aging. We do not yet know what those insights might be, because the deep ocean organisms that carry them have barely been studied. But the potential is there, in the cold and the dark, growing at the rate of micrometers per year. Reproduction in the deep ocean is constrained not just by the slowness of metabolism, but by the fundamental difficulty of finding a mate. In environments where population densities are low and distances between individuals may be hundreds of meters, the problem of finding another member of your own species, specifically a member of the opposite sex that is simultaneously reproductively receptive, is not trivial. At the surface, this problem is solved by visual signals, by acoustic signals, by proximity that comes from living in aggregated schools or territories. In the deep ocean, none of these strategies is reliable. Visual signals fail in the dark. Acoustic signals may propagate long distances but carry limited directional information. Aggregation is rare in environments where food is too sparse to support dense populations. The solution that many deep-sea species have evolved is to produce enormous numbers of eggs and larvae releasing them into the water column where ocean currents carry them over vast distances. This broadcast spawning strategy accepts the low probability that any individual egg will survive, develop, and find suitable habitat, and compensates for that low probability with sheer numbers. A single large female of certain deep-sea fish species may produce millions of eggs in a single reproductive event. The vast majority will be eaten, will settle in unsuitable locations, or will simply fail to develop. But a small fraction will survive, will be carried by currents to locations where conditions are appropriate, and will grow to reproductive age, and the population will persist. The larvae produced by deep-sea broadcast spawning face challenges that surface larvae do not. They must survive in a water column that spans thousands of meters of depth, with different temperatures, pressures, and food availabilities at different levels. They must develop the morphological and physiological adaptations required for deep-sea life during their larval period, before they have settled into their adult habitat. And they must eventually find their way to appropriate adult habitat, a specific depth range, a specific substrate type, a specific chemical environment, in an ocean that offers few landmarks and no visual guidance. How deep-sea larvae navigate to appropriate habitat is not fully understood. Chemical gradients may play a role. Certain substrates and habitats release chemical signatures that larvae can detect and follow. Pressure gradients may provide directional information. The change in pressure with depth is predictable and consistent, and larvae that can sense pressure changes can potentially use them to regulate their depth. Temperature gradients and the acoustic properties of the water may provide additional cues. But the details of larval navigation in the deep ocean remain poorly studied, partly because observing tiny larvae in their natural environment at depth is technically challenging, and partly because the larvae themselves are not easy to collect and maintain in conditions that allow their behavior to be studied. The life history of deep-sea organisms does not end with reproduction. It ends, as all life histories do, with death, and in the deep ocean, death is not merely the end of an individual organism's life. It is the beginning of a process that sustains the community around it. The carcasses of deep-sea organisms, like the whale falls discussed in other contexts, do not simply decompose. They are resources-concentrated deposits of organic matter in an environment where organic matter is chronically scarce. The death of a large organism in the deep ocean attracts scavengers and decomposers that may have traveled considerable distances to reach it, drawn by the chemical signals of decomposition diffusing through the water. Sleeper sharks, slow, patient predators of the deep, are often among the first large arrivals at a carcass, but it is not alone. Mobile scavengers of various kinds, polychaete worms, brittle stars, fish, converge on the site from the surrounding environment. The processing of the carcass by these organisms releases organic compounds into the water and into the sediment, supporting microbial communities that in turn support other organisms. A single carcass can sustain a small ecosystem for months. A brief, intense pulse of biological productivity in an environment otherwise characterized by slow, diffuse scarcity. The recycling of nutrients through deep-sea carcass communities is not merely local. The chemical products of decomposition, dissolved organic compounds, Mineral nutrients released from the bones and tissues diffuse into the surrounding water and are eventually mixed into the broader oceanic nutrient cycle. The organisms that consume the carcass incorporate its organic matter into their own tissues, and when they die or are eaten, that matter is transferred again, moving through the food web in the slow, iterative way that all organic matter moves through ecosystems. The deep ocean has no waste. Everything that enters it is processed repeatedly until it is reduced to its component elements, and those elements eventually cycle back to the surface, where they fuel the photosynthesis that rains organic matter back down again. Life and death in the deep ocean are slower than at the surface, but they are not less complete. The cycles of birth and reproduction and aging and death that characterize every ecosystem on Earth run in the deep ocean at a pace calibrated to the energy available, slow, patient, thorough. The organisms that live there are not struggling to maintain these cycles against the constraints of their environment. They have been shaped by those constraints over hundreds of millions of years into forms that maintain the cycles efficiently and persistently. A deep-sea coral 4,000 years old has outlived civilizations. A fish whose lifespan is measured in centuries has watched the surface world change in ways it cannot perceive. The deep ocean operates on timescales that dwarf human history, and it will continue to do so. The corals will continue to grow, the fish will continue to live their long, cold lives. The larvae will continue to drift through the water column looking for habitat as long as the conditions that make this biology possible remain. What threatens those conditions is not the darkness or the cold or the pressure. Those have been stable for millions of years and will remain stable regardless of what happens on the surface. What threatens them is the surface itself. The warming, the acidification, the changed circulation patterns, the altered chemistry of the water column through which the larvae must navigate and in which the organic matter must rain down. The deep ocean's biological cycles are calibrated to conditions that are changing faster now than they have changed in millions of years. Whether the organisms of the deep ocean can adapt, whether the pace of evolutionary response can match the pace of environmental change, is a question that cannot yet be answered. The deep ocean has survived every previous change that the Earth has inflicted on it, but it has never before been asked to survive a change happening at this speed. The deep ocean's slow time has one more dimension worth understanding: the timescale of evolutionary change itself. At the surface, many species evolve rapidly. Annual plants, insects, bacteria, organisms with short generation times and large populations can accumulate adaptive changes in dozens of generations, which may be as little as a few years. The evolutionary potential of these organisms, their capacity to respond to changing selective pressures through rapid adaptation, is high. Deep-sea organisms, with their long generation times and slow metabolisms, have a different relationship with evolutionary time. An organism that reproduces once per decade accumulates evolutionary change far more slowly than an organism that reproduces dozens of times per year. The population genetics of long-lived, slow-reproducing species favor stability over rapid adaptation. These organisms have been stable in form, in physiology, in ecology for millions of years not because evolution has not acted on them, but because the slow pace of their generation time has meant that evolutionary change accumulates slowly, and the conditions of the deep ocean have been stable enough that there has been no strong selective pressure for rapid change. The coelacanth, a fish long thought to be extinct, rediscovered living in the deep waters off the Comoro Islands in 1938, is the most dramatic example of this evolutionary conservatism. Its body plan has remained essentially unchanged for more than 300 million years. It is not a primitive organism, not a simple organism, not a relic of an earlier and less sophisticated stage of vertebrate evolution. It is a highly sophisticated fish, perfectly adapted to its environment, that has simply had no reason to change because its environment has not changed in ways that required it to. The deep ocean has been stable enough, for long enough, that an organism perfectly adapted to it can persist essentially unchanged for 300 million years. This evolutionary conservatism is both a strength and a vulnerability. The strength is robustness. An organism whose biology has been refined over hundreds of millions of years in stable conditions is extremely well suited to those conditions. The vulnerability is flexibility. An organism whose evolutionary history has not required rapid adaptation may not have the genetic variation, the population size, or the generation time to respond quickly when conditions change. The pace of change in the deep ocean today is, by geological standards, extremely rapid. The temperature of deep water is rising on timescales of decades. The chemistry of the water is shifting on timescales of years. These are timescales on which a species with a 10-year generation time cannot respond through evolution. Natural selection requires multiple generations to act. If the environment changes faster than generations turn over, the population experiences the change as a catastrophe rather than a selective pressure, not an opportunity for adaptation, but a decline in numbers. We do not know the outcome of this experiment. The deep ocean has not experienced forcing of this kind on these timescales in the geological record. What we know from that record is that the deep ocean has survived every past change, including the mass extinctions that periodically reset the surface biosphere. But those changes, catastrophic as they were, unfolded on longer timescales than the current ones and the organisms that survived them, did so partly by evolving in response to the change, and partly by exploiting refugia where conditions remained suitable. The refugia of the future deep ocean, the locations where conditions may remain close to historical norms long enough for organisms to persist, are not yet mapped. They may exist. The deep ocean is vast and heterogeneous, and the rate of change is not uniform across it. Some areas may warm and acidify faster than others. Some habitats may be more buffered against change than others. The organisms of the deep ocean may not face a uniform catastrophe, but a complex, patchy landscape of change, in which some populations decline while others persist, in which some species find refugia while others do not. What emerges from this landscape, on the other side of the coming decades of change, will be a deep ocean community, but perhaps not the one that exists today. Some of the organisms we have described in this documentary may be among those that persist. Others may not. The 4,000-year-old coral may still be growing on its seamount, or it may have been bleached and killed by water that has warmed a degree too far. We do not know. We can only observe and measure and try to understand what is happening before it is over. The deep ocean has witnessed more of Earth's history than any surface environment. It has outlasted the dinosaurs, the ice ages, the formation of the continents. It has absorbed the products of 5 mass extinctions and helped rebuild the biosphere each time. It is patient in a way that human civilization has not yet learned to be. Whether that patience is sufficient for what is coming, whether the slow, deep rhythms of deep ocean biology can persist through the rapid, shallow rhythms of human impact is the most important unanswered question in ocean science. The organisms of the deep ocean are not asking that question. They are simply living, as they have always lived, in the cold and the dark. The question belongs to us. The deep ocean has been conducting experiments in biology for longer than complex life has existed on the surface of the Earth. In its cold, dark, pressurized laboratories, it has tested every variation in protein structure, membrane composition, sensory system design, feeding apparatus, reproductive strategy, and metabolic rate that evolution could generate under its particular set of constraints. The organisms that have survived this testing that we find living today in the deep ocean are not random. They are solutions. Every structure they possess, every biochemical pathway they employ, every behavior they exhibit represents a solution to a problem that the deep ocean posed and that natural selection refined across millions of generations. We have barely begun to read these solutions. The history of deep-sea biology is, in essence, the history of a slowly growing recognition that what lives in the deep ocean is not merely interesting, but useful in ways that extend far beyond marine science into medicine, material science, engineering, and our understanding of the boundaries of life itself. The proteins of deep-sea organisms are perhaps the most immediately applicable example. Enzymes that function at high pressures and low temperatures that remain active and stable in conditions where surface enzymes lose their structure and stop working have direct applications in biotechnology. Many industrial processes require enzymatic reactions at temperatures or pressures that denature conventional enzymes. Deep-sea enzymes, evolved to function in extreme conditions, are natural candidates for these applications. Thermostable DNA polymerases, enzymes that replicate DNA at high temperatures, transformed molecular biology when they were discovered in hot spring bacteria. The cold-active enzymes of deep-sea organisms could do something analogous for processes that require activity at low temperatures, from food processing to pharmaceutical synthesis. Certain deep-sea species produce biological defense materials of extraordinary sophistication, mucus gels that expand explosively in water, consisting of protein fibers thinner than silk with mechanical properties that no engineered material has matched, simultaneously strong, flexible, and capable of absorbing enormous amounts of energy before breaking. Materials scientists studying these deep-sea fibers have found structures that suggest entirely new approaches to high-performance textiles and protective materials. A material with these properties, strong, flexible, lightweight, capable of energy absorption, would be valuable in body armor, in protective sports equipment, in aerospace applications. Some of these organisms have been producing this material for 300 million years. We have been studying it for less than 3 decades. The bioluminescent systems of deep-sea organisms have already transformed biomedical research in ways that most people do not realize. The gene for green fluorescent protein, originally isolated from a shallow water jellyfish, but refined through study of bioluminescent marine organisms, is now one of the most widely used tools in cell biology. Scientists insert the GFP gene into other organisms, causing specific cells or proteins to glow, making them visible under a fluorescent microscope. This technique has been used to track the movement of proteins within cells, to observe the development of embryos in real time, to monitor the activity of neurons in living brains. The 2008 Nobel Prize in Chemistry was awarded for the discovery and development of GFP. The original insight that a marine organism had evolved a molecule that converts chemical energy directly into visible light with near-perfect efficiency came from the study of bioluminescent marine biology. The deep ocean contains thousands of bioluminescent species, most of them unstudied. Each one has evolved its own variant of the bioluminescent chemistry, optimized for its particular ecological context. Some produce light at wavelengths different from GFP. Some produce it more efficiently. Some produce it in response to specific chemical signals that might make them useful as biosensors, organisms, or molecular systems that glow in response to specific environmental compounds. The unexplored bioluminescent diversity of the deep ocean is, in a real sense, an unexplored library of light-producing molecular systems, each potentially useful in ways we have not yet imagined. The venom systems of deep-sea organisms represent another largely unexplored pharmacological resource. Many deep-sea fish, cnidarians, and invertebrates produce venoms, complex mixtures of proteins and peptides that immobilize or kill prey. These venoms have been refined by evolution to interact with specific molecular targets in the nervous systems and muscles of the organisms they are meant to affect. The same properties that make a venom effective as a biological weapon, high specificity for particular molecular targets, high potency, resistance to degradation in seawater, make venom compounds valuable as starting points for drug development. Surface venoms have already yielded important pharmaceuticals. Captopril, used to treat hypertension, was derived from the venom of a Brazilian pit viper. Ziconotide, a powerful painkiller, was derived from the venom of a cone snail. The venoms of deep-sea organisms, operating under different evolutionary pressures, targeting different prey, containing different molecular families, these represent a pharmacological resource that has barely been sampled. The extremophile bacteria and archaea of the deep ocean, the organisms that have evolved to survive and thrive in conditions of extreme temperature, pressure, and chemical hostility, are of particular interest to the biotechnology industry. These organisms have cellular machinery tuned to extreme conditions. Their enzymes are stable where conventional enzymes are not. Their membranes function where conventional membranes fail. Their metabolic pathways process chemicals that most organisms cannot touch. Each of these properties has potential industrial applications, from cleaning up chemical pollutants to manufacturing compounds that are difficult or impossible to produce by conventional chemistry. The deep ocean is, in this sense, a pharmacological and biotechnological frontier, a reservoir of biological solutions to problems that evolution has spent millions of years solving, most of which we have not yet examined. The organisms that contain these solutions are not aware of their value. They are simply living, as they have always lived, in conditions that shape them. But the solutions they carry are real, and the need for them in medicine, in industry, in materials science is real as well. The question is whether we will explore the deep ocean thoroughly enough, carefully enough, and in time to benefit from what it contains. The exploration that has happened so far has produced results that justify the effort many times over. The discovery of thermostable enzymes from hydrothermal vent bacteria transformed molecular biology. The discovery of novel antibiotics from deep-sea fungi and bacteria has provided compounds active against drug-resistant pathogens that surface biology has not produced. The structural analysis of deep-sea proteins has opened new approaches to the design of materials and drugs. Every expedition to the deep ocean that has included careful biological sampling has returned with organisms or compounds that were not previously known, and some fraction of those unknown things have turned out to be useful. The rate of discovery is limited primarily by the rate of exploration. The deep ocean covers more than half the Earth's surface. The biological expeditions that have sampled it systematically, that have gone to specific locations, collected organisms carefully, brought them back alive, or preserved in ways that allow their biology to be studied, have covered a tiny fraction of that surface. The sampling is not merely sparse, it is almost non-existent in many areas. Vast stretches of the deep ocean floor have never been visited by any scientific instrument. The organisms that live there are unknown. Their biology is unknown. Whether they contain useful compounds, novel structures, insights that could transform some field of science, these questions cannot even be asked about organisms that have never been collected. The deep ocean also has something to teach us that is harder to quantify than the value of specific compounds or materials. It has something to teach us about what life is capable of. The organisms we have described in this documentary, the bioluminescent fish with their private light frequencies, the pressure-adapted enzymes that function where surface enzymes fail, the ancient corals that have been growing for 4,000 years, the larvae that navigate thousands of meters of dark water to find habitat, each of these represents a demonstration of what biology can do. Not what biology did once, under some specific past condition, but what biology does routinely, reliably, reproducibly, in conditions that surface life does not experience. This matters for the search for life beyond Earth. The biology of the deep ocean has already transformed. Astrobiology has already demonstrated that life does not require sunlight, that it can function at extreme temperatures and pressures, that it can sustain complex communities in environments that are, by surface standards, uninhabitable. These demonstrations have expanded the range of environments considered potentially habitable on other worlds. The icy moons of the outer solar system, Europa, Enceladus, Titan are candidates for life precisely because deep ocean biology has shown that life can function in conditions similar to those that might exist beneath their surfaces. The next step, actually searching for life in these environments, will require technology that does not yet fully exist. Spacecraft capable of penetrating the ice of Europa, deploying sensors in its subsurface ocean, detecting the chemical signatures of biological activity if it is present. These are engineering challenges of extraordinary difficulty. But they are challenges that scientists and engineers are working on, driven partly by the conviction that the deep ocean's demonstration of life's flexibility makes the search worthwhile. What we find in the deep ocean in the years and decades ahead, as better technology makes more of it accessible, will continue to surprise us. The history of deep-sea biology is a history of surprises, of organisms that should not exist, in conditions that should not support them, doing things that no one predicted. The bioluminescent lure dangling in the dark, the teeth too large for any mouth to close, the 4,000-year-old coral, the male that fuses permanently with the female to avoid the problem of finding her again. Each was a surprise. Each was found because someone went to look. And each turned out to be not merely surprising, but illuminating a window into what biology can do when given extreme conditions and sufficient time. The deep ocean is patient. It has been accumulating its slow wonders for hundreds of millions of years. It does not need us to find them. It does not need us at all. But we need them for the medicines they might yield, for the materials they might inspire, for the understanding they might provide of what life is and what it can endure, for the simple fact that the living world is richer, stranger, and more extraordinary than we have yet managed to appreciate. We have been looking at the deep ocean for less than a century. We have been looking carefully for less than 50 years. And in that time, the deep ocean has changed what we know about life, about evolution, about the conditions under which biology can function, and about where in the universe life might exist. What it will change in the next 50 years as the technology improves, as the expeditions multiply, as the analysis deepens, is not yet known. But if the past is any guide, it will be more than we expect. It always is. The deep ocean is also a model system for understanding symbiosis, the intimate biological partnerships between organisms of different species that, in the deep ocean, have reached forms not found anywhere else on Earth. The partnerships between chemosynthetic bacteria and their animal hosts, tube worms, clams, mussels, represent symbiosis taken to its extreme, not a relationship of benefit and cost, but a merger so complete that neither partner can survive without the other. The bacteria provide nutrition. The animal provides housing, raw materials, and transport. The genomes of both partners have been shaped by the partnership over millions of generations, to the point where the boundary between the two organisms is almost arbitrary. Understanding how these symbioses work, how the partnership is established, how the partners communicate, how the genetic and the metabolic integration is maintained, has implications that extend far beyond marine biology. Symbiosis is a fundamental mechanism of evolution. The mitochondria in every cell of your body are descended from bacteria that were engulfed by a primitive cell more than 1 billion years ago and never released. The chloroplasts in plant cells are descended from photosynthetic bacteria incorporated into a similar partnership. The deep ocean's extreme symbioses are, in a sense, the living descendants of the most consequential biological event in the history of complex life, and studying them may illuminate how that event happened and what made it possible. The deep ocean also offers something that is increasingly rare in biology: a system that has not been substantially disturbed by human activity, or at least a system where the disturbance is recent enough that its baseline state can still be reconstructed. Most of the Earth's surface environments have been altered so profoundly and for so long that their pre-human state can only be inferred. The forests of Europe have been managed and logged for millennia. The coral reefs of the tropics have been fished and polluted for centuries. The grasslands of every continent have been plowed or grazed. Even the open ocean has been affected by fisheries that have reduced the populations of large pelagic predators by 90% or more. The deep ocean, inaccessible to most human activity for most of human history, retains a closer approximation to its natural state than almost any other large ecosystem on Earth. Its baseline is more intact. Its species composition is more similar to what it was before human civilization began to affect it. This makes it scientifically valuable in a specific and irreplaceable way. It is a reference system against which the changes wrought by human activity in other ecosystems can be measured. It is a model of what a large, complex, undisturbed ecosystem looks like, and it is the only such model that exists at global scale. That reference value is being eroded. Deep-sea bottom, trawling, the practice of dragging nets across the seafloor to catch fish, and invertebrates destroys deep-sea habitats on timescales that are essentially instantaneous relative to the timescales on which deep-sea communities recover. A trawl net that passes across a deep-sea coral garden removes organisms that have been growing for centuries and destroys a physical structure that took millennia to build. The recovery of the community, if it occurs at all, may take longer than any monitoring program has yet observed. Deep-sea mining, the extraction of mineral-rich nodules from the abyssal plain and of metal sulfides from hydrothermal vent sites, is a similarly irreversible disturbance. The minerals can be extracted once. The communities that live on them, once destroyed, may not return on any human timescale. The argument for protecting deep-sea ecosystems is not merely aesthetic, though the case on aesthetic grounds alone is substantial. Given what we have described in this documentary, it is practical. The deep ocean contains biological resources, compounds, structures, organisms that we have barely begun to inventory. Destroying the habitat before the inventory is complete is not merely a loss to science. It is a loss to medicine, to materials science, to biotechnology, to every field that stands to benefit from the solutions that deep-sea biology has developed over hundreds of millions of years of evolution. It is also a loss to our understanding of life itself. Every species that goes extinct in the deep ocean before it has been studied takes with it the evolutionary experiments encoded in its genome, the solutions to biological problems that its lineage has been refining for millions of years, the data points in the argument about what life can do and where it can exist. We cannot know in advance which species carry the most important information. We cannot rank the unknown by value. The only way to preserve the option of learning from deep-sea biology is to preserve the organisms that embody it. The deep ocean has given us more in the decades of careful attention we have paid to it than anyone anticipated. It has changed what we know about how energy works in ecosystems, about how life tolerates physical extremes, about how organisms communicate in darkness, about how evolution responds to extreme selective pressure, about where in the universe life might be found. These are not small contributions. They are among the most significant developments in 20th and 21st century biology. What the deep ocean will give us in the years ahead as autonomous vehicles map more of it, as molecular techniques reveal more of its microbial diversity, as pharmaceutical researchers examine more of its chemistry, cannot be predicted. But the pattern of the past suggests that the answers will exceed the questions, that the organisms we find will surprise us, that the biology we encounter will demand revision of what we thought we knew, that the deep ocean, patient and dark and full of things we have not yet seen, will continue to do what it has always done: show us that life is stranger, more capable, and more extraordinary than we had imagined. Rest now. Let the weight of the surface world settle. Below you, in the cold and the dark, organisms that have never seen light are doing what they have always done: hunting, growing, waiting, living in a world that asks nothing of them except that they survive. They have been surviving in that world for longer than we can easily comprehend. And they will continue in that patience and that darkness long after this night has passed. If this documentary moved something in you, if the darkness and the pressure and the patient, extraordinary biology of the deep ocean stayed with you, consider subscribing to Sleep on Deep. Every week, we descend a little further. The ocean is waiting.